Hello students, in the last lecture we saw two general characteristics of chemotherapeutic agents. First is selective toxicity and second one was side effects. So now third, let's see, range of effectiveness. Now drugs vary considerably in their range of effectiveness. So many are narrow spectrum drugs, that is they are effective only against a limited variety of pathogens. Fine. And others are broad spectrum drugs that attack many different kinds of pathogens. So drugs may be classified based on the general microbial group. They act against antibacterial, antifungal, antiprotozoan and antiviral. Okay. Now some agents can be used against more than one group. For example, sulfonamides, they are active against bacteria and some protozoa. Now here I have shown you one table which shows therapeutic drug and its spectrum. So let's see, penicillin, narrow, it is gram positive. Streptomycin, broad spectrum, gram positive, gram negative and mycobacteria. Okay. Bacitracin, narrow spectrum. So same you can see over here, different drugs, cephalosporin, chloramphenicol, ciprofloxacin, erythromycin, sulfonamides, gentamicin and vancomycin. Understood? So this is one more parameter which is range of effectiveness. Next, natural or synthetic agent. Now this chemotherapeutic agents, they can be synthesized by microorganisms or manufactured by chemical procedures which are independent of their microbial activity. A number of the most commonly employed antibiotics are natural, that is totally synthesized by certain bacteria or fungi. So, in contrast, several important chemotherapeutic agents are completely synthetic. Understood? The synthetic antibacterial drugs are sulfonamides, trimethoprim, chloramphenicol, ciprofloxacin, isoniazid and dapsone. Please remember these drugs. Now many antiviral and antiprotozoan drugs, they are synthetic. An increasing number of antibiotics are semi-synthetic. What is semi-synthetic? They are natural antibiotics that have been chemically modified by the addition of extra chemical groups to make them less susceptible to inactivation by pathogens. Now examples, ampicillin, carbanicillin and methicillin. Okay, got it? So this is the table which shows you the source, microorganisms and therapeutic drug. It's given over here in 3 steps: natural, synthetic and semi-synthetic. It's very clear. Okay. Next, fifth parameter is microbicidal or microbistatic. Now therapeutic agents like disinfectants can either be microbicidal or microbistatic. Microbistatic agents, they reversibly inhibit growth. If the agent is removed, the microorganism will recover and grow again. Understood? Simple. And microbicidal agent, it kills the target pathogen. Its activity is concentration dependent and may be only microbistatic at low levels. Fine. It is simple. Okay, you understand both microbistatic, microbicidal. Now microbistatic agents, they do not directly destroy the pathogen. Okay. But elimination of the infection depends on the host's own resistant mechanism. And a microbistatic agent may not be effective if the host resistance is too low. Here I have shown you table which is showing you two steps like microbicidal and microbistatic. Their examples. Okay. Please work on this. Read, write it down. Remember the examples. Next, sixth parameter. It is determination of effectiveness. Now, some idea of the effectiveness of a therapeutic agent against a pathogen can be determined from the minimal inhibitory concentration, MIC. We'll look it in detail later. And next is the minimal bactericidal concentration, MBC. Fine. Now let's see, MIC. It's the lowest concentration of a chemical, usually a drug, which prevents visible growth of bacterium or bacteria. Fine. MIC is also determined by preparing solutions of the chemical in vitro at increasing concentrations, incubating the solutions with separate batches of cultured bacteria. Fine. Next is MBC. MBC is the lowest concentration of an antibacterial agent required to kill a particular bacterium. Fine. Now, a microbicidal drug kills pathogen at levels only 2 to 4 times the MIC, whereas a microbistatic drugs it kills at much higher concentration. Understood? And the MIC results will be like if it is susceptible, intermediate or resistant to a particular antimicrobial agent. Okay. It is often expressed in micrograms per milliliter or milligrams per liter. Okay. Now, one more thing. These results have been graded into susceptible, intermediate, resistant to particular antimicrobial by using a breakpoint. What is the breakpoint? Breakpoints are agreed upon values which are published in guidelines of a reference body such as US or CLSI Clinical Laboratory Standards Institute, the British Society for Antimicrobial Chemotherapy, or the European Committee on Antimicrobial Susceptibility Testing. Fine. So, next, this is the method from which we perform MIC. Okay, there are some dilution steps where we use some solutions. We'll see this in practicals. Okay, then it is incubated for 24 hours and MIC is based on turbidity, so we'll be checking this. So percent concentration of antimicrobial agent will be calculated. Okay. If you have any doubts, do ask me.